holds PhD degree from Naipur Mohali and postdoctoral fellowship from McGill University, Montreal, Canada. Dr. Kumar was specialized in the research areas like oncology, autoimmune disorders, respiratory disorders, and pain. He has published and presented more than 70 papers in reputed Indian and international journals and conferences. Dr. Kumar V.S. Nemmani has over 14 years of industry research expertise on drug discovery, on development, and managerial experience. Now I request Dr. Kumar V.S. Nemmani, Director, Sir, Shri Vishnu College of Pharmacy, to give you a welcome note. Sir, over to Kumar, Sir, Director, Sir, Shri Vishnu College of Pharmacy. Okay, sir, sir is not available due to network issues, sir. Now I will like to introduce today's speaker, Professor Dr. S.H. Ansari, sir. I, Dr. S. Srinivasarao, Coordinator, take this opportunity to welcome today's guest speaker, Professor Dr. S.H. Ansari, sir, to talk about stability testing of herbal drugs, challenges, regulatory complaints, and perspectives. Professor Ansari sir is working as a professor in Department of Pharmacology and Phytochemistry at School of Pharmaceutical Education and Research, Jamia Hamdard University, New Delhi. Sir did his PhD at Jamia Hamdard University, New Delhi in 1997, M.Pharm at Punjab University, Chandigarh, 1981, B.Pharm at Delhi University, Delhi, 1979. Sir also did his BSc HS at International University of Contemporary Studies, Washington, D.C., USA in 2002. Sir is a former Vice Chancellor at Jamia Hamdard University, and also he served in various capacities like Dean, Director cum Dean, HOD, Dean Students Welfare, Chairman of Admission Committee at the same university. Sir is a recipient of several awards and honors to his credit. The most important, the most professional one is Professor M.L. Khurana Memorial Award. For Sir is a former Vice Chancellor at Jamia Hamdard University, and also he served in various capacities like Dean. For best paper in IJPS 2016. He is the visiting professor to a number of universities and organizations at reputed organizations. He is a life member of IPA, APTI, IPGA, IST, etc. He handled research projects worth rupees about 5 crores from different funding agencies like AACT, TST, DPT, etc. He is having 40 years of teaching experience and guided 30 PhD students and number of M.Pharm students. Sir published 2011 research papers in reputed journals. He is an editorial member of several reputed journals. Thank you for giving the opportunity to say about on yourself, sir. Thank you very much, sir. Now I request our speaker, sir, Professor Ansari, sir, to begin his talk. Over to you, sir.
Okay, thank you. Thank you, Dr. Nataraj, Principal, Shri Vishnu College of Pharmacy, and Dr. Srinivasa Rao for giving me opportunity to share some of my experiences in the field of stability of herbal drugs and herbal formulations. As we all of us know that herbal formulation and herbal drugs, the era is always remain under shade because of uncertain studies or uncertainty on quality, efficacy, and safety of herbal drugs and herbal formulations. Unlike allopathic system of medicine, herbal formulations and herbal drugs, they do not have a very good quality parameters. If we talk about quality since last 1 or 2 decades, we are working on quality, we are working on quality standards, the standardization of herbal formulation, herbal drugs. There are various funding agencies which, like CCRUM, Central Council for Research in Unani Medicine, Ayurvedic Council, and other agencies who are giving more stress on standardization of herbal drugs and to establish quality standards of certain plants. National Medical Library includes large number of books on quality standards of herbal drugs. When we talk about efficacy of herbal drugs efficacy, herbal formulations efficacy, which are included in Pharmacopoeia of India, Ayurvedic Pharmacopoeia, Unani Pharmacopoeia. We are working on efficacy, and a lot of work has been done on efficacy. Third important aspect of herbal formulation is safety. Sometimes, very frequently, fluently, we say that herbal drugs, herbal formulations, these are free from side effects, adverse effects. These are safe. But this statement is wrong. Herbal drugs are not always safe. These are safer as compared to allopathic system of medicine. But to say that these are completely from toxicity, they are completely safe from toxicity, this statement is wrong. And stability is a part of safety. When we talk about safety, safety means the toxicity parameters, acute, subacute, and chronic toxicity, and stability also. Product which we are marketing in the pharma formulation, it should be stable. Now I will come to the stability of herbal drugs. What is stability? Stability is the chemical and physical integrity of medicinal products, up to which extent the chemically and physical basis, on physical and chemical basis, any formulation is stable. It is stable means it remains full of active constituents. Their activity remains unchanged with the time. The European Agency for Evaluation of Medicinal Products suggests guidelines on herbal stability testing. So we can say that stability testing is an obligatory requirement in the registration process for all medicinal products, including herbal medicinal products. Next slide, please. Move on to next slide. Yes. The principle of stability is basic principle which is involved in stability testing is to provide evidence on how the quality of an active substance or finished product varies with the time under influence of various environmental factors. For example, temperature, humidity, and light. This is the basic principle that if we increase at accelerated temperature, at low temperature, or under high humidity, low humidity, and light, without light, what will be effect of the active substance, up to which extent the formulation will remain active. This is the basic principle involved. And another principle is to establish retest period for active substance on shelf life of finished product and recommended storage conditions. So when we talk about shelf life, generally we see on herbal formulations label and packages, we see that it is written expiry date is 2 years. But from where this 2 years has been derived, it is not mathematically very much correct or calculated. Other objectives of the stability studies are to evaluate efficacy of drug, for selection of adequate formulation, determination of shelf life storage condition, to develop suitable packaging information for quality, storage, purity, and integrity of product during its shelf life, to provide evidence for regulatory agencies that how quality of drug product varies with time under influence of environment. Any formulation which is to be brought into the market for sale, it is the regulatory requirement that it should comply with the stability studies. Then only it can be sold in the market. Other parameters are the stability studies to help demonstrate the stability of drug substances dosage form, to establish degradation pathways of drug, what are the secondary plant metabolite and basic primary plant metabolite pathways which are affected due to degradation, to elucidate the structure of degradation product, to determine the intrinsic stability of drug. Intrinsic is stability means up to which extent our formulations remain active. They should not lose their activity. Efficacy, treatment of the disease for which these are meant. This is very important that with the time, moisture and temperature, these should remain unchanged. To reveal the degradation mechanisms such as hydrolysis, oxidation, and photolysis. It has been observed that many formulations, when these are stored for longer duration of time, they may go under hydrolysis, oxidation, or photolysis. And finally, to understand the chemical properties of drug formulation. This is the basic principle involved. There are large number of factors which affect drug stability. Some of them are like the interaction between API and the recipient during fermentation process, main formulation. It has been observed that when formulation is stored for longer duration of time, it may undergo fermentation. Many herbal formulations, Unani formulations, they contain sugar as syrup. And if quantity of sugar is less, then fermentation process may take place, which can deteriorate the whole formulation. It also may change physical properties of dosage forms, products, selection of container, closure, packaging system. This is also important with stability. Containers, the type of container, which is it is made up of glass or plastic or metal, that also plays very important role in determining the stability of any formulation. But the most important, as I am telling you again and again, that is effect of storage, that is temperature, humidity, and light for longer duration of time, and selection of solvent system. Which solvent we select for in manufacturing, that is also important. It has been observed that if formulations are prepared by using hydroalcoholic extract or alcohol is used, these types of formulations remain stable for longer duration of time as compared to aqueous extract or where we use water as solvent. And ultimately, if all conditions are not favorable, that may modify the whole formulation. Whole formulation, it may change. Next, please. May I have next slide, please? Yes. This somewhat slightly busy slide. It shows in a comprehensive manner how the mechanism of stability works, functions. Say, as far as stability is concerned of any formulation, the most important is light. Light temperature, at low temperature, higher temperature, the effect of intrinsic value of formulation, it may change. Chemical changes may occur and it may increase or decrease the efficacy of formulation. Due to stability or high after longer duration of time, our formulation, it may undergo polymerization. The active constituents which are present, they may change their structure, they may undergo polymerization. Another important factor is geometric isomerization. Isomers, these geometric or polymeric isomers which are produced after degradation, they may or may not have the biological activity, desired activity. Moisture plays very important role as far as stability is concerned. That's why it is always advisable that we should store our formulation in moisture-free containers, airtight containers. Oxidation process, our formulation, it may undergo oxidation. And finally, hydrolysis may take place, especially the very common example I will give you, the glycosides which are present in any formulation, and if it is not stored properly, it may go under hydrolysis. And you know, when hydrolysis of any glycoside takes place, it produces a glycone and glycone moiety. And if these two moieties are separated, these become inactive. Their activity of glycosides is when these are present together, a glycone and glycone moiety is present in combination. Next, please. May I have next slide, please? Some predictable chemical changes which may take place after long storage are the hygroscopic nature of extract powder leads to flow compressibility problems. If the powders are hygroscopic in nature, they may absorb moisture, and that may affect in its compressibility. Means these cannot be compressed to manufacture tablets, Ayurvedic herbal tablets. Higher moisture content, it may lead great risk of cohesion and adhesion to the containers in which it is used. The presence of variety of phytochemical glycosides, alkaloids, and many other secondary plant metabolites like steroid, terpenoid, etc. These make the formulation very complex and pose challenge for analytical method of development. Means if our formulation undergoes degradation, deterioration, many complex substances, they may be produced, they may occur, and these, it will become a challenge for analyzing these new products which may be harmful for health. Presence of many enzymes like glycosidase, esterase, or oxidases play important role in breakdown of secondary plant metabolites. We know that plant material contains many enzymes, and if under suitable favorable conditions, these enzymes become active and play important role in degradation. This is also a very busy slide which shows possible interactions among different types of phytoconstituents. Some of the examples I have already given, like glycosides. You see, glycosidases enzymes are present in the cells, and if These become active under favorable conditions. These may break the glycone as well as aglycone moiety, and our formulation, it may lose its efficacy. Next slide, please. Similarly, this slide is indicating the heavy metals, etc. There are various types of stability testing. How we can test, how we can perform the stability testing, that is accelerated stability testing, in which product is tested at controlled temperature, humidity, and light, like 40 degrees centigrade at 75% relative humidity. Then another type is real-time long-term stability testing. In this, product is tested for longer duration at 25 to 30 degrees centigrade and 35 to 75% relative humidity, depending upon climatic conditions. At different time intervals, like product is tested for its active constituents after 3 months, 6 months, 9 months, etc. Up to 36 months means up to 3 years. Then comes intermediate testing, in which we test the product at 25 degrees centigrade for longer duration of time. Stress testing, it includes effect of temperature, that is above 40 degrees centigrade and 75% relative humidity. The formulation or product is tested. Forced degradation testing is performed to provide intrinsic stability of drug. Next, please. According to ICH and WHO guidelines, the stability of any product and storage conditions have been summarized here in this table. Some of the examples I will give you. Say room temperature for long-term studies. The test temperature is kept at 22 degrees centigrade plus minus 2 at 60% relative humidity plus minus 5 degree. Similarly, refrigerated at low temperature, it means, and freezer means further at low temperature at minus 20 degrees centigrade. When the product is tested, all conditions have been listed here. Next, please. May I have next slide, please? Please move the slide. Yes. This slide also shows the schedule for stability of new product. If any new product is to be brought into the market, how the various climatic zones can be established. Say for long-term and climatic long-term studies, the zone 1 and zone 4 of climatic conditions can be developed, and environmental condition will remain at 25 degrees centigrade at 60% relative humidity, and product is tested for its active constituents by various analytical methods like HPTLC, etc. after 3, 6, 9, 12, 18, 24, and 36 months. It means the longest study will be of 3 years, 36 months. Then long-term climatic zone 3, it includes 30 degrees centigrade at 35% relative humidity. Again, in this We test the percentage of active constituent in formulation with the help of HPTLC and HPLC after every 3 months interval. Long term for climatic zone 4, for zone 1 and zone 2, condition is 30%, 30 degrees centigrade for 65% relative humidity. Other conditions are enlisted here. Next, please. Now, simply saying that study of stability is simple, but not as simple as we expect. Why it is not simple? Because of very complex nature of herbal formulations. In allopathic system of medicine, since pure constituents, pure substances are used, so it is not difficult. It is easy to study stability of any compound or any formulation in which pure constituents are present. Unlike herbal formulations, in herbal formulations, we have very complex substances. Each and every active constituent is associated with other primary plant metabolites like chlorophyll, alluron grains, starch grains, cellulose, chitin, etc., which are primary plant metabolites and make the formulation very, very complex in nature. And it becomes sometimes difficult to find out exact percentage of active constituent which remains after prolonged storage. So there are various challenges in stability testing, like complex nature I have already discussed. Many herbal compounds are unstable, hence to set to criteria including quality and quantitative parameters. In combined formulation, different substances have significant constituents, give rise to analytical challenges because each and every constituent has its own different analytical procedure for testing. Some constituents, they may have simple procedure, analytical procedure, but there are certain constituents which have very complex and tedious process for its analytical testing. It is not so easy to test by analytical methods. Next, please. Now the testing frequency in stability testing, it is, I can say, repetition of the previous slide, not exactly same way, in which we describe the long-term accelerated and intermediate studies. In long term, after every 3 months, the samples are taken, and every 6 months after 2 years, and finally we test after 3 years. In accelerated studies, the samples are tested at after 3 months, 6 months, and minimum is the 6 months duration for its storage. In intermediate studies, we make 4 points. That is, at 4 stages, we test the stability of any formulation. Say initially, then after 6 months, 9 months, and 12 months. Next, please. Selection of batches and testing conditions had been summarized here in this table, in this slide. Like conducted on at least 3 primary batches. It is important that studies should be minimum 3 batches should be taken, and then average of these 3 batches gives the more accurate results. Next, please. There are some additional parameters, means the parameters which we have already discussed: accelerated initial studies, force studies, etc. Additional parameters which are important in stability testing, which are organoleptic analysis. It is also known as organoleptic properties of any formulation. We should try to find out whether color of formulation is changing after storage or not. It should not change. Color should not change. Odor and taste also of any formulation, it should remain unchanged. There are some physicochemical parameters like pH. pH of formulation or a product, it should remain unchanged. It should not change after prolonged storage. Extractive values, ash values, and insoluble ash values, moisture content, etc. It should not change. After that comes quantitative analysis. Say specific biomarkers, the active constituents present in the plant, which are known as biomarker. Each and every plant has a specific biomarker. It should not change in this sense. It should not get isomerized. Microbiological analysis. This is important because it has been observed that after prolonged storage, there are many formulations which may be infected by microbes. These microbes may be of bacterial or fungal origin. So one has to ensure that the formulation after prolonged storage, it should have not developed microbial growth, bacterial as well as fungal in nature. Next, please. According to ICH and WHO guidelines, in many aspects, stability testing of products require chemically defined substances, means whatever active constituents are present, these should be very well chemically defined. In addition to this, certain factors are to be kept in mind are like herbal drugs and preparations, are active pharmaceutical ingredients. Herbal medicinal products are complex in nature. I have already discussed. Constituents belong to different chemical classes of compounds. Constituents sometimes have very low concentration in the finished product, so it becomes difficult to analyze, to study analytically. It should also be considered the consequences of analysis of herbal medicinal products, like different requirement for different types of extracts. Each and every type of extract has different requirement. I will give you example here. There are some extracts which can be, their active constituent can be tested by HPTLC comfortably. By the same time, there are some extracts in which HPTLC method will not be at much useful. We can take help of HPLC and sometimes TLC also. Fingerprint chromatographic analysis is important sometimes in testing the analytical markers. Next, please. It has been observed that there are some certain limitations or specifications limit, we can say, for special conditions. Limit of assay is permissible 5 point plus minus 5% of declared value. The drugs which have low marker concentrations, the range, it widens to plus minus 10%. Due to influence of in climatic and natural variations, the markers, they also change from extract to extract. Next. Next, please. Since the markers content cannot be defined on a specific level, markers can be defined, but marker concentration, each and every drug has its own level of concentration. Say, atropine. Atropine is present in so many drugs. Concentration, percentage of hyoscyamine and atropine, it varies from drug to drug. Analytical marker is stable to herbal substance monograph is solid dosage form. It remains unstable in dosage form. Many markers, many types of markers we have studied, but the here analytical markers, the markers which are possible to determine analytically comfortably with analytical methods, these markers will be more useful for stability studies rather than chemical markers and biological markers. Next, please. Now, how to find the right marker for analytical? As I told you, analytical, because we are ultimately going to determine percentage of active constituent after various intervals. So, analytical markers will play important role as compared to biological markers, as I told you. Next, please. This slide shows importance of analytical markers in herbal and medicinal plant products. If it is not available commercially, it should be isolated and its structure should be elucidated. Analytical markers, they sometimes enhance the analytical quality control, but always remain unchanged and remain specific for a specific type of formulation. Next, please. I think we should not go much in detail in the case studies. One of my student, he worked on Humulus lupulus of Cannabinaceae family, in which he identified suitable markers, analytical markers, I would like to emphasize here, and which were used for identity testing. So, if analytical markers are not suitable, then alternative markers are searched. So, in this studies, he isolated the markers, established their chemical structures on the basis of various spectral techniques, and the biological activity of that marker was also studied. Next, please. Another example is Tenectum parthenium. He also isolated the analytical marker here, because in stability studies, I am emphasizing again and again, specific marker, presence of specific marker is tested analytically in the formulation after various intervals. To have the knowledge of analytical marker, it is very, very important. Next. Next slide, please. Betula pendula was studied. The spectral data is given here. Next. I think all these are case studies. We will not make. Next, please. So what are various perspectives of stability testing? These are in order to establish safety and therapeutic efficacy of any herbal formulation or herbal drug. This is the perspective we should. There is a need for mandatory evaluation of physical and stability along with major biological activities. It should be compulsory and mandatory that each and every formulation which is going to be sold in the market, these should have comprehensive stability studies. Enzymes that also is suspected remain active, and enzymes play an important role in the formulations, and because of their present pet formulation, it may get deteriorated in a specific time. Next, please. We can summarize the whole process in certain points on the basis of certain points, like herbal medicinal products, just like other drugs, have to fulfill the requirements of regarding quality, including stability testing. This is the legal requirement, mandatory for every formulation. And due to their natural origin, the analytics of all types of extracts is challenged by a complex matrix. Because of very complex nature of herbal formulation, it always remains challenged. In many constituents, in many formulations, the constituents which are present in the extract, these are in very, very low concentration. So sometimes it becomes difficult to test it analytically. Next, please. So finally, we can conclude that herbal and medicinal plant products have number of characteristics. Chemically, analytically defined markers are present in it, and which sometimes may change due to prolonged storage. That indicates the stability, whether the formulation is stable or not at various climatic conditions. And that's what our WHO and ICH guidelines say. Next, please. That's all for this lecture. Once again, I thank Dr. Nataraj, Principal, Shri Vishnu College of Pharmacy, and Dr. Srinivas Rao for giving me opportunity to share some of my views on stability of herbal testing. And this is the area which is still under dark and not very certain, which decides the expiry date of formulation. So before bringing any formulation into the market, we should comprehensively study its stability, and then it should be brought into the market. Thank you very much. Thanks all for your concentration. I am sorry that in the beginning, due to some technical problems, I could not connect myself. But anyway, thank you once again for being here, joining with this. And once again, I thank you all.
Thank you very much, sir. Then the topic is open for discussion. Any audience or participants wish to ask any questions for the Ansari sir, please ask the questions. From the audience, please unmute and ask the questions if any. We are requesting the participants ask some questions to the professor sir. Participants requesting to ask few questions to the Ansari sir about on this topic. Please unmute. Sir, sir, I have one question, sir. Please, sir.
Please go ahead. Please go ahead.
Sir, can you suggest a few journals in which this type of the research was published, sir?
See, specifically, stability studies, stability testing journals with this particular name and title, perhaps these are not available, but in most of the journals which are dealing with natural products, herbal products, the stability studies are Indian Journal of Natural Products, they are publishing, then Chinese Journal of Natural Products and Crude Drugs. These are the journals which are encouraging such type of research articles, and these are published in the journals.
Sir, as per the government of India,
Yes.
they restricted that even the Ayurvedic formulations should be fingerprinted with the biomarkers, sir.
Right. You are
Is
right,
it-
very much.
Is it started in the India market or not, sir? In the Indian research?
These are the regulatory requirements, but it depends up to which extent any manufacturer is following or not. But to my knowledge, it is the legal requirement, mandatory requirement that any formulation, if it is to be brought in the market on international level, then its stability studies and fingerprint marker is must. We can sell it in India, but if one wants to bring any formulation in international market, then these types of studies like fingerprint analysis, complete stability studies, and analytical markers, identification of analytical markers, it becomes necessary for that. It has already been included in regulatory activities in India also. WHO and ICS, they are insisting, but it was not being followed in India, but I am happy that things have started.
Okay, thank you, sir. Actually, I started the fingerprinting analysis in 2018, sir. Unfortunately, this lockdown and the corona effect, my research was backed up, sir. I will again restart my research in this field, sir.
Please go ahead.
Sir.
Fingerprint analysis is important not only for formulation, even for single plant or single drug also you can have the fingerprint analysis.
Yes, sir. Yes, sir. Thank you for your valuable information, sir. Audience, please. Any audience will like it to ask the question? Please unmute and ask the question. Tell me your name, college name, and ask the question.
Sir, I am Aishwarya. I am from Kerala, College of Pharmaceutical Sciences. I am working. I have a doubt regarding the stability studies. Can I ask now?
Yes, please go ahead.
Sir, while performing the PhD studies and all, whether if we are handling with any herbal product, there is any need of any herbal extracts, there is any need of conducting this stability studies?
See, stability studies, as far as herbal extracts are concerned, these stability studies become important when the complete finished formulation is ready. Stability studies, suppose I will give you an example here. Suppose you have a formulation which contains extract of 10, 15, 8, 10 plants, various plants.
Okay.
Study of every extract, stability studies of individual extract will not solve our purpose unless we study the stability studies, parameters of stability studies of finished product. We should give more focus on stability studies of finished products, finished formulation ready to bring into the market rather than individual extract.
Okay, sir. Okay. So that is not mandatory.
Not mandatory, yes.
Okay, sir. Thank you, sir.
Thank you, thank you.
Any participants, please?
Good afternoon. Good afternoon, sir. I am Tanisha.
Good afternoon. I'm not able to hear me. Please speak loudly.
I am Tanuja, sir, from Shri Vishnu College of Pharmacy.
Right.
Sir, can I know what are the qualitative and quantitative parameters that are being involved in the stability testing?
Qualitative parameters, if we talk about quantitative and qualitative, first I will tell you quantitative parameter. Quantitative parameter, analytical markers. Say we have a formulation, we should try to find out percentage of active constituents, which are known as analytical markers of fresh formulation. Then under forced degradation studies, let's say after 3 months, 6 months, 9, 12.
Okay.
Madam, Mathrusri Annapurneswari, madam, are you audible, madam? You have any questions, madam?
We have to find out again its active constituent, and the best method which is used to find out percentage of these quantitative parameters is HPTLC. With the help of high performance thin layer chromatography, we find out percentage of major active constituents. And qualitative parameters, as we discussed in our lecture, that they like color, order, taste, texture, flowability, etc. These are quantitative parameters are very much important because these are directly related with the efficacy of the formulation. Okay.
Okay, sir. Thank you, sir.
Madam Annapurneswari, madam. Mathrusri Annapurneswari, madam, are you audible, madam? Okay, sir. Thank you, sir. Then I will
Thank you very much.
I will like you to give a brief closing note for this today's session. First of all, we will like you to heartfelt thankful to the professor, sir. Even though in his busy schedule, he accepted to deliver a talk and shared his his 40 years of experience knowledge, especially in the stability studies of the health products, herbal health products. Thank you very much, sir. Then sir also discussed about on the need of stability studies, not only for the allopathic, also the need for the herbal formulations. Sir also explained the clear and clear-cut objectives towards the stability studies. And Sir explained the factors as well as mechanisms will affect the stability of drug and its formulations, and predictable chemical changes we may able to guess it during the stability studies. Possible interactions among different types of the phytochemicals, types of stability testing and its protocols, and testing schedules. Thank you for sharing your knowledge to all, sir. Please blessing all the participants, sir. We are all equal to your students, sir. Please bless all the participants, sir. Thank you very much, sir. Thank you, sir. Video. Thank you, Master. Thank you, sir. Okay, sir. I will close the session, sir. Thank you very much. Thank you for the participants. Okay. Sorry.
Sir, thank you, sir. Thank you, sir. I'm sorry, sir.
आज के बाद से जब पहले मैं हाँ करवा दूंगा कि गूगल मीट पे है तभी करेंगे, वरना ये साला पूरा दिन बेकार हो गया काम था।
Sir, principal, sir, I would like to close the session, sir.
Okay, sir. Okay, you can conclude, sir, please.
Thank you, sir. Bye, sir. All the participants, please join on tomorrow's session at 10:45, please. Okay? Thank you all.
No.